So I'm Peter Vranes, CEO, co-founder of Nutromics, and we exist as a company to save millions of lives through continuous diagnostic monitoring. So how are we going to do that? Well, we have a medical wearable device. It looks like that. It's our prototype device, and it has microneedles. And on the tips of those microneedles, we put our DNA-based sensor. And that sensor is a platform technology that can monitor any diagnostic target continuously and in real time. And we also have our launch device, which I have in my hand here, which is a pared-back version of that will be launching in a couple of years. So a few quick facts about Nutromics. We're a medical diagnostics company, world-leading DNA-based sensing technology. We're clinical stage. We've completed our first in-human studies last year. Commercializing in a couple of years. We've raised $35 million. Notable investor is Dexcom. Post-money val of 200 mil on our last round, which was late last year. We have a very large TAM and a world-leading team of 57 based in Melbourne, Australia and San Diego. Let's go to the core problem that we're tackling, and that is that lab diagnostics today provide only a single data point of where a patient was several hours prior. So let's go through how this works. So a test gets ordered. A blood draw is taken, it's sent to the lab where it's processed, and then the clinician gets a single data point of where that patient was when the blood draw took place several hours ago. Now, 70% of all clinical decisions are based on lab diagnostics like this. And for a lot of diagnostic targets that don't move very much, they're quite static, that's fine. But there are hundreds of diagnostic targets that are dynamic, they're changing rapidly. So a clinician doesn't know whether they're trending up, flat, or down. So what do? They have to take another blood draw and they repeat that process. And we know in reality that just simply doesn't happen. It's too expensive, they don't have the time. And unfortunately, that costs lives and it costs billions of dollars in hospital inefficiency. So our solution to the problem is continuous real-time diagnostic monitoring. So our device replaces the blood draw, the sample delivery, the sample analysis, and the single data point. And it's what we call, in effect, a lab on a patch. And it's quite remarkable when you think about all the infrastructure and process that goes into every hospital and in the labs in all those hospitals. We replace that with a simple solid-state device that does it continuously and in real time. Now, we put together a 1-minute animation that kind of puts all this together. So we're in the ICU, patients getting dosed a drug, and we're going to go into the interstitial fluid, which is where we operate, same fluid that CGMs operate in. These are our microneedles, and on the tip of those microneedles, we have our DNA-based sensor. So it looks like a ribbon like that, very selective to its target. So you can see certain targets come along, doesn't respond. Target of interest now comes along, vancomycin. It recognizes that as a shape change, a signal change. We can measure that. It releases it out the left, which is what allows it to be continuous. Each one of those microneedles is a separate sensor. So we've got the world's only wearable tech that can do multi-analyte sensing. Signal goes out through a Bluetooth gateway, out into the cloud, and then back onto pre-existing screens. So a very simplistic way, that's how all of this gets put together. So most of you will be familiar with continuous glucose monitors, $10 billion per year revenue market, growing at double digits, and we are the evolution of that technology. That technology is based on enzymatic sensing, which is not a platform, and our technology, DNA-based sensing, is a platform. So let's have a look at this timeline because it tells a story. So in the last 40 years, there's only ever been 6 enzymatic sensors that have been developed and shown to work on body. Now compare that to what we've done in the last 6 years. We've more than doubled what the whole enzymatic industry took 40 years to do. We did that in less than 6. That's the power of a platform. And then secondly, those 6 enzymatic sensors are all metabolites. So things endogenous in our body. That's the only class of target they can go after. Whereas we don't have that limitation. We can do metabolites, we can do drugs, proteins, hormones. So we can do any diagnostic target continuously and in real time. So it begs the question, if you can monitor anything continuously and in real time, what problems in healthcare are you going to solve? So we've thought long and hard about that over quite some time. And we're starting with therapeutic drug monitoring of the antibiotic vancomycin. So vancomycin is used for sepsis and MRSA. Has a narrow therapeutic window and high toxicity. Very difficult to dose this drug. It's the number one dosed IV antibiotic in the world. 1 in 6 of all inpatients get this drug, so very common. And it's a $3 billion market for us. So you can get a bit of an idea of why we started here. What's the problem? Well, if we go back to the original core problem, delayed and limited data, and it costs lives. So they do what's called trough-based dosing. Typically, you get vancomycin for 7 days, but for the first 2 days, there is no monitoring of the drug whatsoever while they get the patient into what's called steady state. And then after that, they get 2 doses of the drug a day, and they get what's called a trough dose. Now, this is what they actually see. They don't see all those lines continuously. They just see these data points, and they get very little information, and it's delayed. The consequence of that is that up to 43% of patients get an acute kidney injury, and a third of them die. Obviously, clinicians are very worried about toxicity, so they're conservative in their dosing. That has another problem, and that is underdosing. So remember, we're talking about life-threatening bacterial infections here. So underdosing increases hospital stay, and each night in the ICU is another $3,000. So we're going to solve that problem by continuously monitoring with this device, vancomycin, from the first dose. So we'll help clinicians get that patient into the therapeutic range quickly and keep them there outside the toxic range. So this is a video that you're seeing for our clinical trial that we did last year. And this is the application of the patch onto a patient that's receiving vancomycin. And what you're witnessing here is the world's first monitoring of a drug continuously and in real time in a human. So that was a major milestone for us, and we think the industry as well. So we're all about outcomes here. We want to drive to improved outcomes, and those outcomes are a reduction in acute kidney injury. That costs $15,000 on average per patient, shorten hospital stays, and reduce mortality. This year, we're moving into the intended use population in the ICU. So we're doing clinical studies in a number of geographies in the US, Asia, and Europe. And we're also doing clinical studies for device optimization. Now, the patch, as I mentioned before, can do multi-analyte sensing. And we are, in effect, a platform on a platform on a platform. So what do I mean by that? Well, the patch itself is a platform. So as we add additional sensors, we're adding them to the same patch. So we're starting off with vancomycin, going to creatinine and beyond. But it doesn't end there, because the actual electronics in the firmware have been designed for multi-analyte sensing. The sensor itself, we are very vertically integrated. So we can just go from design to a sensor on a body in approximately 4 weeks, very quickly. And the manufacturing process is also designed for multi-product variants. So all of this leads to a really unprecedented ability to have rapid product deployment and go after major revenue opportunities. So how are we going to roll all this out? So we're starting with therapeutic drug monitoring of vancomycin, and then we're going to go further down that drug vertical. Beyond that, we're moving to kidney applications, so sensors for renal. Beyond renal, we're then moving to sepsis, and then the cardiac market. And as you can see here, we're talking about very, very significant TAM opportunities that we'll be tapping into. Funding and milestones. So as I mentioned, we've raised $35 million to date. We've got a $10 million round that will be opening shortly. Then we'll be moving next year to our Series B, which will be circa $30 million. And that'll get us to commercialization in 2 years' time. That'll be outside of the US. And then moving beyond that, we'll be going towards our C round, D round, and then launch in the US in 28. Thank you very much.